Dear students, we are going to study about the topic photosynthesis, which is coming under plant physiology. Photosynthesis is an anabolic process by which green plants prepare its own food materials using carbon dioxide and water in presence of sunlight and chlorophyll to produce carbohydrate or starch. And the equation for photosynthesis is 6 CO2 plus 12 H2O gives C6 H12O6 plus 6 H2O plus 6 O2. From this equation, you would have understood that carbon dioxide is reduced to carbohydrate and oxygen is liberated during photosynthesis. Now we will study the mechanism of photosynthesis. Before telling about the mechanism of photosynthesis, I should mention about the site of photosynthesis. The site of photosynthesis is chloroplast, and in the thylakoid membranes of chloroplast, various pigments are located. The pigments are chlorophyll A, chlorophyll B, xanthophyll and carotene. In this, the chlorophyll A is called as the primary photosynthetic pigment and the remaining three are called accessory pigments. These pigments and proteins are associated on the thylakoid membrane in the form of photosystem 1 and photosystem 2. Going to the mechanism of photosynthesis. The mechanism of photosynthesis can be explained on the basis of two steps. They are light reaction and dark reaction. Light reaction is a photochemical process in which the light energy is harvested by the chlorophyll molecules. And in the light reaction, the assimilatory power ATP and NADPH2, which is required for the dark reaction, is synthesized. One more process in the light reaction. In the light reaction, there will be the evolution of oxygen from water and the process is called photolysis of water. Now moving to the next one, dark reaction. The dark reaction is actually the biochemical phase, and the dark reaction takes place in the stroma of the chloroplast. I would have mentioned that the light reaction is taking place in the grana or the thylakoid membranes of the chloroplast. Now once again I am stressing that the dark reaction is taking place in the stroma of the chloroplast. And in the dark reaction, using the assimilatory power ATP and NADPH2 or NADPH, the carbon dioxide is reduced to carbohydrate. Light reaction. Light reaction involves four steps. The first one is photoexcitation of chlorophyll molecule. The second one is electron transport chain. The third one is photophosphorylation. And the fourth one is photolysis of water. There are two types of photophosphorylation. The first one is non-cyclic photophosphorylation and the second one is cyclic photophosphorylation. Coming to non-cyclic photophosphorylation. The non-cyclic photophosphorylation, it includes both PS1 and PS2. At the same time, one more process is taking place, that is photolysis of water. In the non-cyclic photophosphorylation, there is the production of ATP molecules as well as NADPH. We will explain the non-cyclic photophosphorylation. In the non-cyclic photophosphorylation, the PS2 absorbs the light energy and the reaction center of PS2, that is P680, ejects the electrons, and this electron passes to an electron acceptor, and from the electron acceptor it goes through a series of electron transporters and finally it reaches the PS1 or the pigment system or photosystem 1. During the transport of electrons through the electron transport system or electron transport chain, there will be the production of one ATP molecule. The PS1 also will accept the light energy, and the PS1, the reaction center P700, also will be ejecting the electrons, and the electrons, it moves to the electron acceptor, or the electrons will be accepted by the electron acceptor. And we will come back to this PS2 once again. What is happening here? In the PS2, there is, with the oxygen evolving complex, one more process will be taking place, that is the photolysis of water. Water will be splitting into H plus ions, electrons and oxygen. By this photolysis of water, the electrons produced will move to the PS2 and the electron gap of PS2 will be filled by the electrons from the photolysis of water. And the H plus ions produced here will be utilized for the production of NADPH from NADP with the help of PS1. Now, cyclic photophosphorylation. In the cyclic photophosphorylation, PS1 absorbs the light energy and the reaction center of PS1, that is P700, will expel the electron. The expelled electrons will be accepted by the electron acceptor. After that, it moves through a series of electron transporters and finally it comes back to PS1 again. During the transport of electrons through these electron carriers, there will be the production of ATP molecules from ADP and inorganic phosphate. Now I will explain why this phosphorylation is called as cyclic photophosphorylation. In the cyclic photophosphorylation, the electrons expelled from PS1 is coming back to PS1. That means the path of electrons here, it is cyclic. So this is called as cyclic photophosphorylation. In the cyclic photophosphorylation, there is the production of ATP molecules, but there is no NADPH production. Now you would have understood that why this non-cyclic photophosphorylation here, the electrons expelled from PS2 reaches the PS1 and the PS2 is getting the electrons from the photolysis of water. That means the path of electrons here, it is non-cyclic. That's why it is called as non-cyclic photophosphorylation. Dear students, we will discuss about the dark reaction. In the dark reaction, the carbon dioxide will be reduced to carbohydrate with the help of assimilatory pair produced during the light reaction. Which are the assimilatory pairs? ATP and NADPH2. And the fixation of carbon dioxide in dark reaction, it takes place by two methods. One is C3 cycle and another one is C4 cycle. C3 cycle is also called as Calvin cycle. C4 cycle is also called as Hatch-Slack pathway. We will discuss about the C3 cycle, which is a common cycle in all the green plants. In the C3 cycle, the primary acceptor of carbon dioxide is RuBp, and you know about RuBp. RuBp is ribulose 1,5 biphosphate. RuBp combines with carbon dioxide to produce a 3-carbon compound phosphoglyceric acid. And the first stable product of this dark reaction is 3 carbon compound phosphoglyceric acid. That is why we call this cycle as C3 cycle. Now 3 phosphoglyceric acid, it will be again phosphorylated to 1,3-diphosphoglyceric acid. And this 1,3-diphosphoglyceric acid is reduced to phosphoglyceraldehyde, 3-phosphoglyceraldehyde. Some of them will be isomerized to form dihydroxyacetone phosphate. And now this phosphoglyceraldehyde and dihydroxyacetone phosphate, which are both 3 carbon compounds, fuse to form a 6 carbon compound fructose 1,6-diphosphate. Fructose 1,6-diphosphate will be converted to fructose 6-phosphate, again changing to glucose 6-phosphate. Glucose and glucose will converted to sucrose and finally to starch. Now we understood how the starch is produced or carbohydrate is produced through dark reaction. The next step is regeneration of RUBP. For the regeneration of RUBP, phosphoglyceraldehyde molecules, some of the phosphoglyceraldehyde molecules, it again through a series of reactions, it forms RUMP, ribulose monophosphate, which is again a 5 carbon compound. This ribulose monophosphate will be phosphorylated to produce RUBP or ribulose biphosphate. Now, let me talk about the utilization of ATP and NADPH in this reaction or in this cycle. Here, for the phosphorylation of 3-PGA to 1,3-DPGA, ATP molecules will be utilized. Similarly, for the phosphorylation of RUMP to RUBP, again the ATP molecules will be utilized. Now, where is NADPH molecules are utilized? The NADPH molecules are utilized during the conversion of or the reduction of 1,3-DPGA diphosphoglyceric acid into phosphoglyceraldehyde. So, the steps in the dark reaction or C3 cycle can be divided into 3 phases. The first one is carboxylation, the second one is reduction, and the third one is regeneration, that is regeneration of RUBP. Dear students, we will move on to the multiple choice questions from the chapter photosynthesis. The question number 1. Synthesis of complex organic substances from simple inorganic raw materials in the presence of sunlight and chlorophyll is called as dash, which is a dash process. Now, you can go through the various choices. Option A is photosynthesis anabolic. Option B is photosynthesis catabolic. Option C, respiration anabolic. Option D, respiration catabolic. Now, before solving this one, I want you to recollect about anabolism and catabolism. What is meant by anabolism or anabolic reactions? Anabolic reactions are the reactions in which complex organic molecules are prepared from simple inorganic substances. And catabolic reactions. Catabolic reactions are the reactions where complex organic molecules are broken down into simple inorganic substances. Can you give some examples for anabolic reactions and catabolic reactions? Anabolic reactions. Protein synthesis, it is an anabolic reaction because from small amino acid molecules, large protein molecules are produced. Similarly, photosynthesis, here also using the simple raw materials, carbon dioxide and water, complex organic material is produced, that is starch. Now, catabolic reactions, which example you have studied? You have studied about respiration, cellular respiration. The breaking down of complex food material into simple inorganic substances. You would have got the answer. The correct option is photosynthesis, which is an anabolic process. Dear students, let us discuss the next question. The next question is, accessory photosynthetic pigments in most green plants are options A. Chlorophyll A, B. Chlorophyll B, C. Carotenoids and xanthophylls, D. Both B and C. Now let us discuss what is meant by accessory photosynthetic pigments. Accessory photosynthetic pigments are the pigments which help in photosynthesis, and you know about the accessory photosynthetic pigments which trap the sunlight as well as which will transfer the energy captured to the primary reaction center or the chlorophyll A molecule. And in most of the green plants, the accessory pigments are chlorophyll B, xanthophylls and carotenoids. Now, what is chlorophyll A's role in the photosynthesis? Chlorophyll A's role in the photosynthesis is the starting or the initiation of photochemical process. So, chlorophyll A is the primary photosynthetic pigment and chlorophyll B, xanthophylls and carotene, these are accessory photosynthetic pigments. So, which option should be correct here? The option D, both B and C, since chlorophyll B, xanthophylls and carotene, all these they just trap the sunlight and pass it to chlorophyll A. So, they are called as accessory pigments. The next question, if green plant cells are incubated with oxygen 18 labeled carbon dioxide, which of the following molecules will become radioactive when the cells are exposed to light? The options are option A, ATP, option B, water, option C, sugar, option D, oxygen. Now students, let us recall the equation for photosynthesis. The equation for photosynthesis is 6 CO2 plus 12 H2O in presence of sunlight and chlorophyll gives C6H12O6 plus 6 H2O plus 6 O2. From this equation it is very clear that the carbon dioxide, one of the raw material, is reduced to carbohydrate. So, if the labeled oxygen is there in the carbon dioxide, among this, which compound will be radioactive? Definitely it will be C6H12O6. That means what it is? It is the sugar. So, which option should be correct in this? Option C, sugar is the correct answer. The next question is, dash is the process of synthesis of ATP from ADP and inorganic phosphate in presence of light. The options are option A, phosphorylation, option B, photophosphorylation, option C, photosystem, option D, oxidative phosphorylation. Now, let me ask you, what is meant by phosphorylation? Phosphorylation is nothing but the formation of ATP molecule from ADP and inorganic phosphate. And we know that there are three types of phosphorylation. Which are the three types? One is which is taking place during the photosynthesis, that is photophosphorylation. Since the phosphorylation is an endothermic reaction, in this phosphorylation the light is providing the energy. So, this phosphorylation we call it as photophosphorylation. Now, the next one I want to ask you, what is meant by oxidative phosphorylation? Oxidative phosphorylation, it is taking place during the cellular respiration. That is, here also the phosphorylation takes place, but here the energy comes from the oxidation of reduced coenzymes. Now, what is meant by substrate level phosphorylation? Of course, that is not there in the option, but substrate level phosphorylation, it also takes place during respiration. At what stage of respiration? In Krebs cycle. When Succinyl coenzyme A is oxidized to succinic acid. At that time, when one substrate is changing into another substrate, the phosphorylation takes place with the released energy, and this is called substrate level phosphorylation. So now you have understood what is phosphorylation, what is oxidative phosphorylation, what is photophosphorylation, what is substrate level phosphorylation. So you can answer, which will be the answer? The option B, photophosphorylation will be the correct answer, because in photosynthesis, light is providing energy for the phosphorylation. Students, the next question is, photochemical phase does not include, and the options are option A, light absorption, B, water splitting and oxygen release, C, ATP and NADPH formation, D, carbon dioxide fixation. Let us recollect what is meant by photochemical phase. Which phase of photosynthesis is called as photochemical phase? It is the light reaction. Now, what happens in the light reaction? In the light reaction, there will be light absorption, then there will be photolysis of water, and there will be the formation of ATP and NADPH. Where does this carbon dioxide fixation occurs? The carbon dioxide fixation, it occurs in the stroma of the chloroplast, and where it is connected to the dark reaction. So, which is the correct answer here? See, the first three options, light absorption is taking place during the light reaction, water splitting and oxygen release is also taking place during the light reaction, ATP and NADPH formation is also taking during the light reaction. Carbon dioxide fixation is not taking during the light reaction, so carbon dioxide fixation is the correct answer. So option D is the correct answer. Now the next question is, refer the given reaction. 2H2O in presence of sunlight produces 4H plus oxygen plus 4 electrons. Where does this reaction takes place in the chloroplast of plants? Options are option A, outer surface of the thylakoid membrane, option B, inner surface of the thylakoid membrane, option C, in the matrix or stroma, option D, intermembrane space. Now, students, which is this reaction? This reaction is actually the photolysis of water. What is photolysis of water? It is the light induced splitting of water. I want you to recall the non-cyclic photophosphorylation here. In the non-cyclic photophosphorylation, how the electron gap of PS2 is filled? It is filled by the photolysis of water. What is associated with the photolysis of water? PS2. Let me ask, where is the location of PS2? The PS2 is located on the inner surface of the thylakoid membrane. So, where will be the site of the reaction? The site of the reaction will be on the inner surface of the thylakoid membrane. What will be located on the outer surface of the thylakoid membrane? The photosystem 1 is located on the outer surface of the thylakoid membrane. PS2 will be located on the inner surface of the thylakoid membrane. What is the role of matrix or stroma here? The stroma is a site of dark reaction. What is the role of intermembrane space? Intermembrane space is maintaining the hydrogen ion concentration, or it is involved or it is concerned with the ATP production or chemiosmosis. So, which option will be correct here, students? The option B, inner surface of the thylakoid membrane, is the correct answer. The next question is, in C4 plants, Calvin cycle enzymes are present in option A, chloroplast of mesophyll cells, option B, chloroplast of bundle sheath cells, Option C, cytoplasm of gut cells. Option D, cytoplasm of epidermal cells. Students, I want you to recollect about the Hatch and Slack pathway what you have studied. I want you to recall about the C4 cycle seen in C4 plants. C4 cycle, C4 plants, it shows a special type of anatomy which is called as Kranz anatomy. And what is meant by Kranz anatomy? This is a special type of anatomy seen in C4 plants, and this Kranz anatomy, there are two types of chloroplast cells in mesophyll cells as well as bundle sheath. The chloroplast of mesophyll cells, they are normal chloroplast, and the mesophyll cells possess the enzyme for the carbon dioxide fixation. And the bundle sheath cells, they possess a granule chloroplast, and bundle sheath cells actually possess the enzymes for carbon dioxide fixation by Calvin cycle. So, from this it is very clear that the initial carbon dioxide fixation is taking place in mesophyll cells, and then second step, the carbon dioxide fixation by Calvin cycle is taking place in bundle sheath cells. So, where will be the enzyme for the Calvin cycle in C4 plants? The enzyme for the Calvin cycle will be definitely present in the bundle sheath cells, because in the bundle sheath cells, by the decarboxylation of malic acid, the carbon dioxide concentration gradually increases, and that triggers the Calvin cycle. So, definitely the enzymes for the Calvin cycle will be located in the bundle sheath cells. So, which option will be correct here? Option B, chloroplast of bundle sheath cells. Already we have mentioned which enzyme will be present in the mesophyll cells. PEP carboxylase enzyme will be present in the mesophyll cells. Now, C and D, that is cytoplasm of gut cells and cytoplasm of epidermal cells, both are not at all connected with the fixation of carbon dioxide. One, cytoplasm of epidermal cells, and the function of epidermal cell is protection. And gut cells, it will be mainly helping in the opening and closing of the stomata, that means main process transpiration. So, the option B is the correct answer. Select the incorrect pair. Option A, 2-carbon compound aspartic acid. Option B, 3-carbon compound phosphoglyceric acid. Option C, 4-carbon compound malic acid, and option D, 5-carbon compound RUBP. Let us see how many carbon molecules are present in each of these compounds. I will start from the last option, that is option D. RUBP, how many carbon molecules are present? You know that it is the initial carbon dioxide acceptor in C3 cycle, and it is a 5-carbon compound. Now, the next one is malic acid, and the malic acid, you know that it is a 4-carbon compound and it is produced during the C4 cycle. Now, going to the next one, previous one, that is PGA, phosphoglyceric acid. And you know that the phosphoglyceric acid, it is the first stable product of Calvin cycle or C3 cycle. So, definitely it is a 3-carbon compound. Now, coming to the aspartic acid. Now, aspartic acid is also there in the C4 cycle, and aspartic acid is also a 4-carbon compound. It is very clear that phosphoglyceric acid is a 3-carbon compound, malic acid is a 4-carbon compound, RUBP is a 5-carbon compound. At the same time, aspartic acid is a 4-carbon compound. Which is the incorrect pair here? The option A, 2-carbon compound aspartic acid is the incorrect pair. Other three, option B, C and D, PGA is a 3-carbon compound, malic acid is a 4-carbon compound, RUBP is a 5-carbon compound. next question is, identify the correct sequence of stages of Calvin cycle. Option A, reduction, carboxylation, regeneration. Option B, carboxylation, regeneration, reduction. Option C, carboxylation, reduction, regeneration. Option D, reduction, regeneration and carboxylation. Now students, I want you to recall the Calvin cycle. In the Calvin cycle, initially there will be the formation of 3-carbon compound phosphoglyceric acid by accepting carbon dioxide molecule by RUBP. And you know that process is called as carboxylation, addition of carbon dioxide, formation of an acid, carboxylic acid, carboxylation. Now, what happens to the PGA produced here or phosphoglyceric acid produced here? The phosphoglyceric acid produced here, it will be further reduced to phosphoglyceraldehyde, which will be further reduced to glucose, and that step is taking place with the utilization of NADPH produced during the light reaction. And we know that NADPH is a reducing power, and that particular step, that means the formation of glucose through a series of reaction from PGA is a reduction. Then the next step, that is the formation of RUBP via RUMP. Some molecules of PGAL produced will be utilized for the regeneration of RUBP, and that step is called as regeneration. So, the correct sequence of the reactions in Calvin cycle is first one carboxylation, formation of phosphoglyceric acid. The second one is reduction, formation of glucose, and the third one is the regeneration, the formation of RUBP or the regeneration of RUBP. So, which will be the correct option here? It should be carboxylation, then it should be the reduction, then it should be the regeneration. So, students, which will be the correct option? The option C is the correct option, that is carboxylation, reduction and the regeneration. I think I don't have to explain about the other three options. Dear students, the next question is, how many ATP and NADPH2 are respectively produced in the process of photorespiration? Options are option A 2 and 4, option B 1 and 2, option C 4 and 6, option D 0 and 0. Now, let me ask you what is meant by photorespiration, where it is taking place and when it is taking place. Photorespiration is actually a wasteful process where there is no formation of sugar, where there is utilization of ATP and there is no production of NADPH. Then where it is taking place? It is taking place in C3 plants. What time it is taking place? It is taking place when the concentration of carbon dioxide is low, when the temperature as well as sunlight is more. Why it is taking place? At this time, the normal enzyme, carbon dioxide fixing enzyme Rubisco, it is actually catalyzing the combination of RUBP with oxygen instead of carbon dioxide. That means it is mainly showing oxygenase activity, and as a result, what is produced? As a result, one molecule of 3-carbon compound phosphoglyceric acid is produced and one molecule of phosphoglycolate is produced. Now, here there is no production of NADPH, there is no production of sugar. At the same time, there will be the utilization of ATP molecule produced. See, what is in the question? How many ATP molecules are produced? How many NADPH2 molecules are produced? Is there the ATP molecule production? No. Instead, what will be there? ATP utilization will be there. Now, is there the production of NADPH molecule? Nothing. There is no production of NADPH molecules. And which is the correct option? The option D 0 and 0, since there is no ATP production and no NADPH2 production.